Thank you, Lex. And yeah, thank you, Lex and Doreen, for inviting me here today, for organizing this. I think this is an extremely important topic, obviously. And I'm so proud to be here in the Netherlands, which I feel is one of the countries that takes research integrity very seriously and is trying to do something about it. As I have moved across the ocean, I've had some experience with lots of countries, and I feel the Netherlands is one of the leaders in this aspect to make this a topic that is discussable and that is just an important part of science, as it should be. So I have this sort of dual personality. I started my career as a microbiologist, and I am now a science integrity consultant, so I'm not longer employed, I'm self-employed. I run 2 blogs. Microbiome Digest is the one that I sort of started my online career with, and hence my Twitter handle, Microbiome Digest without the E. But currently I'm more involved in science integrity. I just cannot change my Twitter handle anymore because I would lose all my followers, and I think that would be, I would really miss them. So I'll talk here today about a lot of things that could lead me into trouble. And if you follow me on Twitter, you know I'll be regularly attacked for all the types of problems that I've raised. And so I would like to start with my disclosure summary, because I'm asked the question a lot, like, who is paying you? And I am a consultant, so I am paid by universities, by publishers, sometimes by lawyers, to give talks about this topic or to look at particular cases. I also have 4 patents. This is from the time that I worked at a startup called Biomedical. And that company is now, the founders are being charged with fraud. So I don't think these 4 patents are ever going to generate any income for me. I also have a Patreon account, which is a site where people can donate $1 or $5 a month. And this is not, I'm not saying this because I want money, but this is a source of income for me. And these are just people who want to support my work and don't have any strings attached. I'm not being paid by particular people to investigate the cases that you will see me talk about online. All the consulting work I do is not public. I cannot disclose my clients. But all the work I do on Papier or on Twitter, that is the work that I do unpaid. And that is basically because of my Patreon account, I can afford to do all of that work. And so I also want to acknowledge a lot of people who do very similar work as I do. So I'm maybe the person who does a lot of this under my own name. And as you can imagine, the work that I do is not always, doesn't make me a lot of friends. So as Boris already alluded to, a lot of people do this work under a pseudonym. And some of these people are also on Twitter. And we do have a network of people who, where we discuss particular papers or where we ask opinions about particular figures. And I want to thank all of these people, a lot of them are listed here, or the people who provide the platforms that we can use, like Papier. So I do want to acknowledge that some of the work I'll be discussing today has not been done by me, but has been done by some of these other people who mainly want to work anonymously. So why do all of us care about science integrity? Because science is built on publications. Every science publication builds upon the work of previous scientists. So we publish our work, we cite other people's work, we hope that our papers will be cited by others. All of us researchers base our research on the work of others. So I see science as a brick wall in which every brick rests upon a layer of bricks below that. And if one of those bricks, one of those papers contains an error or contains fraud, that means that all the other papers built upon that paper are not on stable ground. And so when a paper contains a problem or a suspicion of fraud, we need to discuss that, and we need to make that a discussion, and we need to give feedback and warn others about that. That is why we are so, why we feel that this work is so important. So most scientists are honest and hardworking, and we might all make errors. I don't want you to leave my talk thinking that all scientists are fraudulent. That's not the case. But as a field, science is not immune to fraud either. So science misconduct could be all kinds of things. So in the United States, we have the Office of Research Integrity that defines science misconduct as one of three things: plagiarism, so copying somebody else's ideas or quotes without giving credit. There's falsification, where people might have obtained results, but tweak them, maybe leave out an outlier, maybe increase a value a little bit or decrease it, basically changing some of the results to make them better fit your hypothesis. And then fabrication is where a person might make up results completely, doesn't do an experiment, just sits behind their computer, types in some numbers and creates a beautiful graph without actually having obtained any measurement in the lab. So I also want to point out that behind every case of misconduct, I think there's a sad story. There's a story of people who felt that the only way to get further in their career is by cheating. People who feel an enormous amount of pressure to publish. People who might work in a lab where there's a bully as a professor who threatens to fire them. Or people who have obtained beautiful results as a postdoc, had high expectations, then became a professor, but their research now is not really going as well as it used to do. And they feel they need to live up to all the promises. And they feel they can only get that by cheating. So when I talk about misconduct, I sometimes have to name and shame people a little bit, but I do want to make it about the figures or the data. I don't want to make it about the people. I don't want to criticize people too much. I don't want to look up where they live or how they look like or comment on that. I think we should focus on the science and the problems that have arisen with those papers. So those are some important things. And when we think about scientific papers and the problems that we can discuss maybe on papier in these papers, there's just an enormous range of all kinds of problems. There could be things like plagiarism or errors or control groups that are not okay. So I focus, and I'm most famous for finding duplications in figures in scientific papers. But I've also worked on all kinds of other problems that you might find in these papers, and not all of these are science misconduct. In fact, there's a huge sort of gray area where a person can either do a very perfect paper without any criticism, or they can do flat-out misconduct. But there's actually a huge gray zone in between, where you can think of all kinds of research malpractices, maybe picking the wrong statistical error, but not doing that really maliciously. But yeah, there can all be all kinds of other problems. And all of these problems should be discussable, and not all of these problems are misconduct. So I specialize in research images. And when you think about scientific papers, most papers actually don't have any photos. Most papers will just have line graphs or tables or sequence data. But when papers do photos, and especially biomedical papers, photos might look like the ones you see behind me. They might be photos of tissues or cells or blots or bands or gels, things like that. And when you look at these images, each of these photos is unique. Like almost everything in nature, if you compare a bunch of leaves or a bunch of faces or a bunch of rocks to each other, we can tell them apart. We can look at them and recognize faces. I'm actually very bad in faces, so I would not recognize any of you tomorrow. But I can recognize photos. And most humans have this ability to look at these photos and recognize that even though these bands are all similar to each other, we can tell them apart. Each band, for example, in the top right, if you look at these, this is a Western blot, you can see all these bands that all have slightly different shapes and gray values and thickness and slants, but they're all different. We can tell them apart. So each of these photos are fine. There's nothing wrong with them. I have looked at them for many times. I have not found any duplications here. But there could be all kinds of problems in scientific papers, And I like to show this sort of simplified example. So these are not images you would typically find in a scientific paper. These are images of sunsets in Hawaii or somewhere else, somewhere nice. And you can see, this is just to illustrate the certain types of problems you could see with papers that have figures. So I don't recognize a good Photoshop. I would totally be convinced that it's a real photo. But I can look at images and see duplications. And I think about half of the people in a typical company can actually see that. So if you look at the category 1 type of duplication that you see on the left, you'll see a bunch of photos, and you'll see that 2 of these images are identical to each other. And I put a green box around them. So this is a very easy to spot duplication. It's the same photo, the exact same photo is used twice. And in a research paper, it will be used twice to represent the same, to represent 2 different experiments. And that's not okay. This is an inappropriate image duplication. And I call this category 1, a simple one. In the middle part of the slide, you can see a category 2 duplication. Here you can see 2 images that have an overlap with each other. You can see part of the image is the same, but the image has been shifted. So this is a duplication with a repositioning. I call this a category 2 duplication. It could also be mirroring, zooming in or zooming out, or rotation, things like that. If you then look at the example on the right, which I call a category 3 duplication, this is actually my very bad attempt at photoshopping. You can see that one of the images has the same palm tree twice. And that's a duplication within a photo. Now, if you can sort of consider these 3 categories, I think it's fair to say that category 1 is most likely to be an honest error, while category 3 is very unlikely to be an honest error. And category 2 is somewhere in the middle. And this categorization helps me to decide what would be the best way to correct or to retract a paper, how to act upon this problem. So I've looked at many scientific papers and categorized them into these 3 images. And none of them was of palm trees, unfortunately, but they would look like this. So here's an example from a scientific paper in which I've shown you a simple duplication. You might look at this for a long time and maybe you will spot it immediately or not. But there's a couple of panels here that are identical, even though they represent different experiments. And I don't see these things immediately. I have to look at it for a while, and then I might see a duplication there. And I've marked it on the next slide. So there's 2 panels here that, if you look at the labels, they're different, but it's more or less the same photo. It's the exact same photo used twice to represent different experiments. So this is a simple duplication. And this could be an honest error. Somebody just had a bunch of these Western blots, didn't label them correctly, which is not good research practices, but it's also not maybe done with the intention to mislead. Although it might be, I mean, I don't know. But this is a category 1 error. So I've reported this to the journal, and I've reported this in October 2015 to PLOS ONE. And PLOS ONE has not addressed this. Well, this is maybe not really a very big error. You might not really care about this too much. But still, this is a paper cited by 106 people. And there's an error here, which is pretty obvious, but it hasn't been addressed. Here's an example of a type 2 duplication. So here's an example where you see 4 different photos, 4 different experiments. Each of these represent a different amount of radiation. So cells were treated with different amount of radiations. There's 4 different irradiation versions. So each of these 4 photos should be an independent experiment. But if you look at this for a while, you might see that some of these photos overlap. And we would expect, based on the labels, that there were 4 different photos. But 2 of these, the top 2 ones, overlap with each other, marked by a green box. And the right 2 ones also overlap with each other, marked by a dark blue box. So instead of looking at 4 photos, we're only looking at 2. There's some overlaps here. It's not just a simple overlap either. So it is a duplication with repositioning, a type 2 duplication. But there's also some change in aspect height. The height width is different. And there's a zoom in and zoom out. So I think this is less likely to have been an honest error. And something went wrong with the text here, but this paper got corrected. This was also in +1. I think this should have been, personally, I would have liked this to have been retracted. I think if an author has 4 experiments and labels things so, so sloppily, then that you don't know which photo belonged to what experiment anymore, I would not trust any other results from that lab either. And there were actually 2 of these figures in the paper. But this resulted at least in a correction. The authors were allowed to send in a new set of photos, and the journal accepted that. Here's another example of a type 2 duplication, where you can see a repositioning. And maybe you already spotted it. The one on the top left is quite easy to recognize. So here are two panels, 2 Western blot panels that represent different proteins. They're called BACs and BAD, and it's bad indeed because one of the photos appears to have been mirrored, flipped. And but it's the same photo if you look at it carefully. And there's another set of panels, the 2 GAP-DH panels, also seem to be the same. Might be a little bit harder to see, but they're stretched differently in the vertical axis. These are the same photos as well. So there's 2 duplications here of the type 2. And this paper, I cannot read there's something wrong with the font, so I cannot really read it, but I think this wasn't addressed yet. This paper I did report to the journal, and nothing happened. Here's an example from a famous lab in France that is not very happy with the things I found. Here's a type 3 duplication. Here's one photo of what we call a southern blot, so it's a blot of DNA, not protein. And at first glance, this photo looks fine. There's lots of different things to be seen here, and all the bands seem at different heights, and they all slightly look differently. But if you look at it with maybe specifically looking for duplications, you might have spotted that several of these parts in this image look very similar to each other, as I call unexpectedly similar. And I've marked them here with colored boxes, and you can actually see 4 different duplications here. I cannot think of any technical explanation that could account for this, but I've posted this on Papier, and unfortunately nothing has happened yet. This is also a pretty popular paper. It's cited by 116 other papers, and the author has not responded to this comment that I posted on Papier. Here's another example of a type 3 duplication. So again, a duplication within the same photo. Well, actually, this is not a photo. This is a graph. It's a line graph, I guess. But the human eye can still look at all these dots, and you can see that you can have spotted that there's repetitive patterns that are very unexpected here. So again, a type 3 duplication. This is a flow cytometry plot. I call these flaw cytometry plots because there's something really bad going on here. Maybe you've spotted it already. There's lots of duplications here. There's duplicated parts within the same panel and across different panels. I don't think this is real at all. Maybe the middle one was real, but the other ones were sort of generated out of Frankenstein parts of the other plots or so. But yeah, this is a plot that appears to have been manipulated. So I've reported this on Papier. I haven't reported this to the journal yet, but because that takes a lot of time. And yeah, nothing has happened yet. But I would not trust anything from this lab. So I've shown you some examples. A couple of years ago, I did this sort of as my hobby. I was still fully employed, and at night and in the weekends, just for fun, because I enjoy doing this, I looked for images for duplications in biomedical papers. And I turned this into sort of a big hobby, and I actually scanned 20,000 papers by eye, spanning 20 different years, spanning 40 different journals from 14 different publishers. And if you think that yeah, that should have taken years to do. Well, it sort of took me a couple of years to do because I still had a full-time job. But I only looked for the images. I don't read these papers. I just scan for them, and I can do one a minute if it only has a couple of images. Some papers, some journals have very complex images, complex figure panels, and they might take up to 15 minutes. But I can do, yeah, simple papers I can do pretty fast. So in that set of 20,000, I found 800 papers to contain duplicated images, like the ones I've shown you in the previous slides. That's 4%. So there were these three types, the simple, the repositioned, and the altered, these three categories that I talked about. So that sort of helped me to make to answer the question, how many of these papers are manipulated? What is the percentage of misconduct in the papers that I've scanned? So just looking at these photos or these flow cytometry images, about half of them I would estimate to have been altered with the intention to mislead the reader. And it's hard to say it for the individual paper, but because we had these three categories, that's our best educated guess, that about half these papers have been, the photos in these papers have been duplicated with the intention to mislead the reader. So does that mean that 2% of all papers contains misconduct? I think it's actually much higher. I think it's around 5 to 10%. And that actually matches the recent survey that has been done, in which, very dependent on the field, I think it was about between 7 and 10% of people who admitted that they might have done misconduct in their papers. And why is it much higher than the 2% that I found? That is, I'm just looking at photos. And it's typically easy to see these duplications if you can see them, or at least once you point them out. But how hard would it be for me to detect if a table was manipulated? I would not be able to detect that. Or a line graph. Anybody can draw a very convincing line graph. I would not be able to spot that that was completely made up, as long as you do it well. I can really only catch the tip, the very tip of the iceberg. The people who are dumb enough to leave traces. The equivalent of the burglar who leaves their driver license next to the stolen television. Like really, these are, for me, fairly easy to catch. I'm actually worried that the real amount of, the real percentage of misconduct is much higher than 2% and might be in the between 5 and 10%. And maybe even higher, because probably not everybody is going to admit that they did that even in a survey. And I also had a chance, because we had this nice data set, to have to sort of look at a relationship between the impact factor of a journal and the percentage of problems that we found in these papers. So I looked at 40 different journals, and this is the graph in which I tried to correlate the impact factor, which is on the x-axis, and the percentage of problematic images of papers, which is on the y-axis. And you can sort of see that there's an inverse relationship between them. If in the bottom right, you see journals with very high impact factor, that was Nature and Science, and I did find image duplications in those papers as well, including type 3 categories. So those, in general, they had a low percentage of these problems, but they still had some problems. Some journals with very low impact factor, I'm looking at Spandidos in particular, they have these journals have very high percentage of problems. But there's also some journals with low impact factors that do really, really well, that have very low percentage of problems. So it's probably dependent on the editorial team, the staff that works at these journals, or maybe the volunteers who run these journals, that determine not only how well these papers are screened, but probably also what type of audience they attract. Maybe the people publishing in Science and Nature are very well experienced researchers who know how to do research misconduct really well. This could be an interesting hypothesis. And maybe that's why we see a low percentage. And of course, there's more professional people looking at a paper that is published in Nature or Science than a paper that is published in a low impact journal in general, generalizing here. But there seems to be an inverse relationship, and I think these are important things to discuss and to know. But yeah, it's not a 100% correlation. I also know a little bit about how journals respond, and we already sort of discussed this previously. How well or how poorly do journals respond? So that set of 800 papers that I reported all of these to the journals, and by now this is about 5 or 6 years ago. If you focus on the left bar in this graph, those are all 782 papers that I reported 6 years ago. And unfortunately, after waiting for 6 years, which is a generous time to give to a journal to respond to my complaints, 60% of these papers had not been acted. They're still out there with their duplication. Nothing has happened, just crickets. About 28% of these papers have been corrected. And about 10% of these papers, after 5, 6 years, have been retracted. I think this should have been half. But, you know, it's nice that papers are corrected and retracted, but I think it's freaking bad that, given the amount of money that the scientific publishing business make, that 60% of these papers has not been acted upon. I've written to these journals under my full name, still with my Stanford affiliation, in the hope that it would give some credibility. But in most cases, journals didn't even respond. Or they said, we'll look into this, and then nothing happened. So, yeah, that's pretty bad. I tried to play it according to the rules, reporting it to the journal editors, keeping it quiet, not rocking any boats, but the journals didn't really respond. And so, just showing some bad editorial decisions here. Here we have a photo in which I have found, let's say, some problems. There are some duplicated elements here. This is a transmission electron microscopy photo. I've never done TEM, but I think something is really bad going on with this photo. And so I wrote to the journal, and the journal allowed a correction. They allowed the researcher to send in a new photo that had no problems, or was just a better Photoshop, I don't know. And yeah, it got corrected, problem solved. Well, the big problem here is that the author is actually an associate editor of that journal. And so you can sort of see that the conflict of interest that might play a role here. The main editor, the editor-in-chief, might not have, you know, might have been best buddies with the associate editor, at least they know each other. And yeah, they didn't seem to want to really slap this person too hard on the hand. They just gave him the chance to correct this image. And I think this should have been a retraction. If you would follow COPE guidelines, this is a manipulated photo. I think this should be 100% retraction. There's no doubt about it. So this is what I call the bad editorial decision, and I'll be happy to post this online on Twitter and start a discussion. Here we have another bad editorial decision. This is what I would call the naive editor. If you look at these images, you see lots of parts that appear to have been cloned, again, a type 3 duplication. And yeah, I sent this in, and the authors then sent in another set of photos, and they said, oh, yeah, there were some incorrect blots sent. Well, yeah, incorrect, but it's, in my opinion, this appears to have been photoshopped. So here the editor just was very gullible and just accepted the new set of photos. And this paper was also corrected, where it should have been retracted. So lots of these examples. I have about 4,000 papers that I've reported now, so I could show you many more. But the point is that I tried to play it in the official, professional way. I contacted the editor-in-chief. I contacted the research integrity officer of the university. They might have started an investigation or not. But the fact is that the majority of these cases are not acted upon. And I think 1 year should be a reasonable amount. I've waited even 5 or 6 years. So I gave everybody a chance to respond, but nothing happens. So I'm now doing it the sort of frustrated and proactive way. I'm posting on Papier, because I don't really care that much about what happens to these papers or the authors. But I want to warn the people who might use these papers, who might base their research on these papers. I feel they need to be warned. We need to have a quick method of telling people that there might be a big problem with this paper. And so I'm posting on papier.com, and I'm posting on social media. And some people might call that trial by Twitter. in which I try to maybe insinuate that people did misconduct. And of course, I have a lot of thoughts, and I've shared some of them with you today there about those cases. But in many cases, I try to post on Twitter just showing the problem, and then not saying this is misconduct. But of course, people will then draw their own conclusions. And sometimes it helps to discuss these things on social media. We know about, for example, the Me Too movement, about sexual harassment cases, which are obviously of a very different proportion than scientific misconduct. But sometimes it helps to bring these cases out in the open and discuss them online. And it might force journals or institutions to act. But my main purpose is to warn other readers. And so I've been a power user of Papier, and I can recommend the plugin. And I've shown you also here on the right side of the slide, you see this example of what if you do a particular literature search, you might see these green little banners showing that these papers have a Papier command. And you can click on them and see what people had to say about these papers. And so, obviously, the work that I'm doing is ruffling some feathers. I'm making lots of enemies because I'm criticizing other people's work. And not only do I do that, there's other people doing that as well. So here are 3 examples in which I have not been involved. I haven't really been involved in these cases, of 3 researchers who have been criticized on Papier or somewhere else, and who, instead of answering the comments on Papier, they decided to threaten with legal actions. So some people discussed some papers about social spiders, and there were big problems in these papers that were found. But instead of these papers being swiftly retracted, they're still under investigation because the main researcher who has been accused of misconduct is threatening with legal action. Or there have been cases where there were many comments on Papier, and that the researcher wanted to sue Papier, but that luckily didn't happen, but there were definitely threats. There have been articles for example, in the New York Times, in which the facts were presented, but still the researcher tried to sue the New York Times for publishing a paper about his potential misconduct. So these legal threats can be quite scary, and they can be quite intimidating. And I don't think they're really meant to win the case. They're meant to silence the people who criticize these people's work. And so I've been involved in a case myself. There was a famous paper. We just heard that President Trump tweeted about this paper. I think the only time he tweeted about a scientific paper at all. And it was this paper, and the paper claimed that hydroxychloroquine was the treatment of choice for COVID-19 cases. It was based on a very small patient set, and it had lots and lots of problems. I wrote a long critique about it on Papier. I wrote a blog post about it. I found lots and lots of problems in it. I wasn't against hydroxychloroquine. I just didn't think that there was enough evidence in this paper to switch completely to hydroxychloroquine as the method of treatment of choice. So a couple of weeks or so after this paper was discussed, not just by me, but others as well, an expression of concern was put on the paper, but then it disappeared again. And it's still a bit unclear. Like, this paper is still, it's not retracted, even though there have been many other papers showing that this paper could not possibly have been true. And why did this expression of concern disappear? So there was actually a journalism report where they showed that the editor or the publisher had received a threatening letter by the senior author in which he threatened with legal action if this expression of concern did not quickly disappear. And so it did. And so it seems that the editor or the publisher decided to not pursue this expression of concern or investigation because one of the authors threatened to sue him. And so this is very intimidating if you get a letter like that. And I can comment on that myself. I've received also a letter like that. So this professor from the hydroxychloroquine is threatening to sue me. There was also another case in which I received a very intimidating legal letter from a completely different author in which they asked me to remove blog posts, to remove tweets, to remove Papier posts. And I wrote them back saying, I'm not going to do that. And then they wrote me back saying, well, that's not very wise. But then it stopped. So I hope it's just a threat. But I'm not being really sure about that. And it does keep me up at night because, I don't know, I'm just an independent person. I don't have Stanford University backing me up with powerful lawyers or the New York Times, who has a whole legal team. I'm just me. And this could financially ruin me. But I'm willing to do that. I'm willing to fight that fight. And luckily, I'm supported by a lot of people in the scientific community who think the same way, who think that scientific discussions do not belong in the courtroom, but belong amongst scientists themselves. That all these issues that we raise on Papier could be answered. All these concerns could be taken away by the authors themselves, because maybe they have a very convincing explanation for some of the problems that we're finding. Maybe some of the problems, for example, with permits or IRB approvals that were not mentioned in the paper. Maybe they were really there. Maybe they just got mentioned in the paper, but maybe the author could convince me that really the research had been done under the appropriate approval. So I think in many cases I can raise concerns, but the author could also reply and take away these concerns. When they don't, when they choose to not reply, but choose to sue me, I think I've actually asked the right questions. And so this strengthens me to keep on asking these questions. But again, I'm very supported by this outpour of the scientific community. And I'm very proud that on the same day I appeared both in Science as well in Nature. I don't think I would have ever been able to do that with my regular research. And it's just fantastic to know how much support I've gotten for these cases. So I still have some time left. So I've talked a lot about duplicated images. Most of these cases are, I believe, from sort of individuals who cheat, who want to cheat for whatever reason. But there's also organized crime in scientific publishing, and those are paper mills. And paper mills are companies that sell scientific papers that are completely fake to authors who need them. And this is a particular problem in China, not because people in China are more or less fraudulent than people in other countries, but because there's a specific incentive, a specific requirement in China for medical doctors who are very early career, who have finished medical school, and who want to work in a hospital. They have the requirement to publish a scientific paper. Now, in the US, you can choose, if you do a medical school, you can choose to have a completely clinical path or to choose a research career or something of a hybrid, but you're given time off to do research. We all know we cannot just do research in our free time unless you're crazy enough like me to scan papers in your free time. Most people don't have the time to do an additional research project on top of their regular work. And especially if you're a medical doctor and you do an 80-hour clinical shift every week, you don't have the time to do research. You might not even work in a hospital that has research facilities. And so how do you fulfill this impossible requirement? You buy a paper because it's an investment for your career. Once you have your paper, once you can check that box, you can forget about research. You might not care about research at all. You just want to focus on patient care. And so these medical doctors massively buy these papers that are completely fake. We think that they're fake. They look real, though. The papers themselves look surprisingly real. You would not really see the pattern. It's only when people like us who scan massive amounts of papers, we start to see patterns. And we found these papers that appear to all have been written on the same template. They all seem to be the same paper, but they have slightly different topics. And you wouldn't catch any plagiarism in there. There's not a single sentence. In most cases, there's not a single sentence that's the same. But yet, we discovered, for example, a group of papers that we now call the tadpole paper mill. And I'm not sure if you can all read it, but their titles were surprisingly similar. If you sort of start to see a pattern, and it's almost like some random generator chooses from a set of words and generates a new title, but based on this very given structure. And similarly, the structure of the papers was also very similar. But most importantly, so here are two examples of papers. This is a different paper mill. We call this the stock photo paper mill. This is a group of 125 papers that I've worked on. And in these papers, at first glance, these papers look not that similar to each other. They have different figures. They're both retracted now. But if you look very carefully and have some memory for photos, you might see these two images that I've marked here with a red box. It doesn't really appear here very well. These photos are actually the same. But these are two different papers, two different sets of authors, but they use the same photo. How can two sets of very different authors that are not related to each other use the same photo? Well, because they bought this paper from the same paper mill. So if you look at these photos in more detail, you can see they're from two different papers. They're supposed to represent very different experiments. And yet, these photos are the same. They're zoomed in differently. They're upside down with respect to each other. But if you see this little area that are marked with the letter J, you might spot that these photos are exactly the same. And so they were sold to two different groups of authors. There's actually 125 papers that all pulled from the same set of papers, same set of photos. And then there's other paper mills that we found. Here's what I call the comb paper mill. And these have correlation plots that all look very, very similar to each other. You might spot that all these four plots, they come from 4 different papers, 4 completely different sets of authors, no overlap, 4 different hospitals, 4 different provinces in China. But the graphs are remarkably the same. They all have very similar dots. Some dots were removed, some dots were added, but it's all based on the same template. And so you cannot really see this if you look at these as an individual paper. You might not spot that. Excuse me. But if you compare all of these and start to see the patterns, these papers all look very similar to each other. And then here's one that is also from a paper mill. And not sure if you can read it, but this is an example of why we think that this paper comes from a paper mill and that they're all written on the same template. So we actually found this set of papers because they all used a very similar text as the legend of Table 1. And this particular paper talked about prostate cancer patients. And I don't know if you've spotted that about half of the prostate cancer patients were female. And I don't think this paper was really peer-reviewed. And once I point this out to you, you might never look at a paper again the same way. But there were many papers in this paper mill. Some were prostate cancer. You expect a certain gender ratio there. But some were colon cancer, or breast cancer, or liver cancer, or colon cancer. And they all had this same table, but all the numbers are different. You would not be able to find this by just scanning for the numbers, but they all had a very similar text in the table. The legends were all very similar, which is how we were able to find them. And of course, by pointing out these things on papier, I'm also telling the paper mills how to not do it the next time. So they'll be better, they'll be scanning for these things, and I believe by now we won't be able to recognize paper mill papers again. I think at this point we've reached a point where we can no longer know if a paper is real or not. Maybe only can confirm that by replicating the results. So I'm going to skip these, and I'm going to end my talk with a discussion. So this is a slide usually where I end with by asking some questions to think about it. So I've talked a little bit about why people do misconduct. And again, these are sad stories. I don't want to ridiculize people because I know there's often cases where there might be a bully as a professor, a bully as a PI, and there might be a graduate student who feels the only way out of that lab is by producing particular results. And if you have a professor who says, I'm going to fire you unless you have this paper ready by the end of the month, what are you going to do? In academia, we're so dependent, there's so much hierarchy, there's so much power of the PI that as as a lower member, a junior researcher, you're often intimidated by that. And because I'm Dutch as well, I know I'm not very intimidated by bosses. But in most other countries, that is the case. The boss is the boss, and he or she can fire you. And if you're on a visa in the US and you're fired, that means you have to leave the US within 5 days. You have to pack up and leave the country. That is super intimidating, especially if you have your family and you have to go back to another country. You can sort of see that people might be desperate and might just think, I want to get out of this lab. I'm going to fake some results as long as I get my paper. So I do think we focus way too much on publications and productivity as a measure of productivity. Not sure how to change this system, but I sort of want to plug for more measures of reproducibility. And I wish there was a type of research job in which people could just reproduce other people's results. I think a lot of researchers would be totally happy with that as a career, especially people who love working in the lab, who don't want to manage other people, who don't want to think about getting grants, but just replicating other people's results. I think that's the only way we know if a paper was real or not, because if you think of how easy it is to generate fake videos with, I don't know, Tom Cruise on golf courses, and it's not really Tom Cruise, how easy would it be to make a fake Western blot? Very easy. And so we would not spot that. The only way, I think, of knowing if a paper is real is to replicate it. We've touched a little bit upon the many conflict of interests that are within publishers and institutions to not investigate these cases. Unfortunately, I've had too many emails where clear cases of photoshopping were judged by the institution to be, something went wrong in that lab. We fired the postdoc, but the professor is still there and actually gets there another NIH grant the next year. And I feel we need to hold the people in power positions more accountable, and not just fire the poor postdoc who felt they needed to do this, but to also focus on the atmosphere in every lab. Whose role is it to detect science misconduct? So I'm mostly an unpaid volunteer, sponsored by Patreon, but I'm not getting paid for this by a salary. I do feel there should be more effort within the publishers, who we pay hundreds of millions of dollars to get our papers published. We write our papers for free, we peer review them for free, we might even edit them for free, and yet we have to pay $4,000 to get a paper published. Where does that money go to? I'm not always sure about the answer of that, but I feel there should be some effort. If you sell a product that is so expensive, you need to have some quality control, and you need to be held accountable. If I buy a car and the wheel falls off a year later, I feel I have some rights as a customer. And so if we find an error in a scientific paper, there should be some recall mechanism where the publisher should be held accountable for the quality. I also hope there's better legal protection for whistleblowers like me, or just in general for scientists who raise concerns about pandemics or origin of viruses. How can we better be protected from the influence of social media, the harassment of people online, or even the threats to our family? There should be better measures taken for that, because most scientists want to tell the truth, and we fight for that, and we should not be punished for that. And in the end, I think the most important thing to remember is not only does science misconduct have a tremendous cost for science in general, because we might base our research on results that didn't happen, but also for the scientific community in general at all, because there's already so much misinformation, there's already so much distrust by the general audience in science papers, that all these bad papers that are getting out there and published, that then later get retracted or discussed, that even ruins the trust of the audience in science even more. And I think as a scientific community, we need to be better guardians of the quality in science and to act faster if we find a faulty product. So with that, thank you for your attention. You can follow me on Twitter. I'm Microbiome Digest. You can play the game Image Forensics. I make it about a game there, not about the paper. And you can win an emoji award if you are the first person to find the duplication. Thank you.
Well, thank you very much, Elisabeth, for this fascinating and also sobering lecture. I learned a few more new things, and they were not all nice, I should say. And specifically, I find it so disappointing, like you say, that these journals respond so badly. You would imagine that the only thing they really sell is quality control. And it's amazing that they don't have their acts together. So I'm really puzzled. I'm really puzzled. And they do have problems in that industry with getting that act together, amazingly. Might it be, what are your thoughts on that? That they're so feared for lawsuits that that's the reason they respond not or so slow?
That might be one of the concerns. I also think that most bigger publishers have grown into this waterhead of managers, and I don't even know, like shiny buildings that do, I'm not quite sure what, lots of people who do wonderful things, I'm sure, who talk about policies, but not actually, people on the floor who address these problems. PLOS One, for example, I've been to PLOS, that has a beautiful shiny office, very expensive in San Francisco. And it was the first time I was at the publisher. I'm like, what are all these people doing here? Because I'm peer reviewing these papers. And then, it's like some automated system. And then like what are all these folks doing? I don't know. But, we pay them money, but that doesn't need to cost that many, or shouldn't have that many people. And yet, there's this shiny building full of people who do something, but all these papers that I put on their lap, they didn't have time. They said, oh, we don't have staff to look at these things and to contact the authors. I'm like, well, what do you do then the whole day? They actually hired 2 people to deal with the 400 papers that I dumped on their lap. So they do now about 1 paper a week of the 400 that I sent them. So it will take them, I don't know, 20 more years or so to handle all these cases. And I've been sending them new ones as well. So I have a very pessimistic view maybe on scientific publishing, and maybe I'm, stepping on some toes here, but I do feel we need to go to a new model, and we need to have, maybe send less money to these publishers and think about new ways of publishing results that could also count towards our careers and our resumes. Maybe publish just 1 figure. That's one of the things I'm thinking of, where you publish 1 figure, I get points for that, other people replicate this, they also get points for that, and then we have all together proven that this is a scientific experiment worth building our careers on. But I hope we can sort of abandon the very costly scientific publishing model, make it much more open and transparent.
Yeah. Well, thank you very much. There's a question there. Please go ahead.
Bas van Steensel, Netherlands Cancer Institute. The few times that I've contacted journals or an institute about an allegation, a problem, I found it took me so much time to do it right. But you have to write the perfect letter, you have to collect all the evidence, triple check everything. How do you do this? Because it's so time consuming. Is there a trick, a tip that you have how to limit? Because it's unfair, right? Somebody else made a mistake, and then you as a whistleblower have to put in so much effort.
Right.
So what is the trick here to deal with that?
I wish all the journals would subscribe to Papier and spend a tiny bit of money and have an automatic email sent to them, like, some of your papers have been commented on, here are the problems, done with it. It's so much work, like you said, for me to write to, I've by now 4,000 or 5,000 papers that I found. And most of them I haven't even reported. The 600, the 800 I did, but later on I sort of posted online. It's too much work to even find the email addresses of the editors, because they're very hidden on most journals. If I want to contact a research integrity officer, that can sometimes take hours to find, to hunt down, because they have a lot of beautiful policies online, but no person to contact to. Or I cannot contact the university because I don't have the university email address, so I don't have access to their sites. It's only made for internal whistleblowers. And so I actually have a database that we worked on with several whistleblowers with all the email addresses that we could find. But even that, it's hard to keep track, especially when I send an email to 20, let's say I have a set of 23 papers, which were published in 20 different journals. every journal has like maybe a journal email address and an editor-in-chief and 3 other editors-in-chief. It takes me like 4 or 5 hours to gather all that information, to send all the papier links to them. And then some might respond, some might not respond. I cannot keep track of that. That would be a full-time administrative job, and I don't have time. So my administration is not very good. And I'm sort of more and more abandoning the idea of even sending this to the publishers.
Yeah. There are many questions online, and I take 2 simple ones for a start. How do you select the papers, the journals you're going to scan?
I work off many leads, so I feel like I'm a detective. I get anonymous tips, I get emails, LinkedIn, tweets, direct messages. And I work from the leads that I still have based on that initial set of 800, because some of these papers I want to check out other papers by the same authors. Or I read about a case online, I'm like, oh, let's check it out. So yeah, many different ways that I get information.
Yeah, multiple angles. Yeah. So we can send a tweet to you and then you might respond.
I might respond. I get more every day than I can handle. And yeah, it's too much. It's too much.
The other simple question is, or maybe not so simple, I'm not sure. You do commissioned work as well. And when you find something ugly, do you post that on papier as well? No,
I cannot do that because that's the work I do as a contractor. That's, I guess I have to keep that confidential. Yeah. So usually I have to sign something that I cannot
Agreement
post
of confidentiality.
Yes, yes. There might be some cases in which actually a person might want to publish it and might ask me to post it on Twitter or papier, but all the cases I've worked on under such contract, I do privately. So I just send them a report back, and it might actually be in favor of the person, might be as a defense for that person.
Sure. Yeah, that's an option as well, of course. All right. Another question from the room. Zico.
You just called yourself a detective. I actually think you're more of a Dark Knight. You're not the Dark Knight we deserve, but the one we need. You're basically Batman. And Batman does his job as a vigilante because nobody else is doing it. And that's why I wanted to follow up on one of your suggestions just now on doing more replication studies. One idea I've heard a couple of times over the past years is that wouldn't it be a great idea for every PhD course or trajectory to include at least one replication study?
Yes.
Because then we can all become Batman. What do you think about that suggestion?
Well, I don't see myself really as Batman because I feel I just do work that anybody could do. And I don't have any superpowers. But yeah, I think that's a wonderful suggestion. And I think, you know, how cool is it to replicate somebody else's results? You'll learn a lot. You'll learn how to read a paper. You'll learn how to, you know, hold a pipette and how to read between the lines and how to know what things were actually not mentioned in the methods, which are many things. We all know that if you work with RNA, it can be, you know, if you shake something for 200 RPM, is that like an orbital shaker? Is it back and forth? That could make a big difference, and that is often not mentioned in the methods. And so I think it's a wonderful way to learn how to not write a paper and how to write it in a paper that, how to write it in a way that is replicable for other people as well. And I think a lot of people would love that as a first check, especially when you're in your first uncertain years and you have no idea how to do an experiment. Just replicate somebody else's results and get credit for that.
Yeah. Well, one more question from the room, please.
First of all, thank you very much for doing this really important and rather thankless job. And I was wondering, you said there was a correlation between impact factor and the amount of doctored images. And was there also a correlation between the impact factor and how they would reply to your you pointing that out?
Well, no. I think it's very dependent on the journal. I've actually had very bad experiences with Science and Nature. And it's very heavily dependent on the case, so which makes me suspect that in some cases there are huge conflicts of interest. When a young researcher, a postdoc or a graduate student can be thrown under the bus, that will happen and the paper might get retracted. Or when maybe it's a not native or English-speaking person, they get thrown under the bus also. So there's also potential racism there. it's very dependent on the case, and I'm planning to write a paper, but I never have time because I get so many requests for new cases, to see what happened to these 800 papers, and correlate that to impact factor, open access journal, other editors, I'm not quite sure what, but like I think there's many conclusions to be drawn from that. But from my first impression, it's very dependent and seems to be very random what happens to a paper.
Yeah, we even get anonymous questions online, which is good, and Papier is well, inspiring this maybe even. And this is an interesting question maybe as well, and that is, you said, I'm focusing on the work, on the integrity of the work, and not on the person. But at the same time, you mentioned several times that you wouldn't trust anything from this lab when something bad occurred in one paper of that lab. Is that consistent with each other?
Yeah, that sounds maybe contradictory. What I was trying to say is that online, I try not to ridiculize people. I do think you can draw your own conclusions when you search for a particular name and you see that that particular author had 40 Papier comments. I think you should be able to draw on your own conclusion, especially if you see that the older papers, that person was first author and then sort of moved, and that all the papers with that name as last author also has problems. I do think I have opinions, obviously, about people, but I try not to really mention that too much online, because I know also that that could break a person's career, and I'm not always 100% certain who was the culprit. Like usually papers have many authors, and if you just look at one paper, I wouldn't trust anything from that paper if there was a Photoshop. I just think that one Photoshopped image should be enough to retract the whole paper and to put serious concerns with other papers from that lab. But it's hard to know which particular person that is from. So I think it's more on a lab level than on a personal level where you can draw these conclusions, although there are exceptions to that.
Yeah, sure. That's clarifying. Yuri, the microphone, please, because people online need to listen to it as well.
Well, thank you so much for this beautiful talk and the beautiful, outrageous work that you're doing. It's beautiful to see how much effort you put in this. I had two questions. The follow-up question is, maybe I misunderstood this, but I think you're not contacting the corresponding author, right? And I was wondering, why are you not contacting them? Because if I was running a lab and I was the corresponding author and the first or the second author did this, I would really want to know first to have a discussion with the author and retract it myself. So I would feel so much more inclined to do that. So that's my first question. Yeah.
So the answer is that in most cases that's not very productive. I do indirectly contact the authors through Papier, because when you post a problem about a paper and you make sure that the email of that author is added to the Papier, they will get a comment, they will get a message that there's a comment on your paper and you can reply on that. I think there, because it's online, it's public, the authors usually don't say something bad. If I would directly contact an author, I'm in some cases where things went wrong and the editor actually forwarded my whole email, including my email address, to the author. That was in the beginning. The author was very angry with me, and I didn't feel that was very productive, because they can send me angry emails back because it's not in the public eye, so they will be less inhibited to say something bad about me. So
I would send a grateful email back to say.
I have received very few thankful emails on Papier.
Yeah, okay.
And the thing is that most of these things are not honest errors. I do believe most of this is misconduct or very sloppy.
Yeah.
And when I see comments on Papier, very often, don't have any numbers, they're negative. They're trying to drag my whole past, all my papers, or anything they can find about me personally, so they counterattack. And it's very rare that I've seen responses where the senior author says, wait, this is serious? There are a couple of exceptions, I've seen that. But and I just know by the reply of the author if they were involved in a misconduct or not. Because if they say, what you're saying, I'm gonna look into this, that's very serious, I do believe they had no idea. And but if they say, you're a bad woman, then and they sort of clap back to me, I feel I've yeah, I've hit them where it hurts.
Yeah, I understand. Can I ask another question?
Yeah, short one.
Well, it's about passion. So what makes you continue this work? I mean, all these nasty emails, all these nasty tweets, the threats, the everything, and the hard work. I mean, it costs so much time. So what? And of course, the quest for honesty is an important supporting fact, motivating factor, maybe. But why are you continuing this work?
I think, and this sounds, of course, very arrogant, but I believe I'm right. I ask the right questions. I have the right to raise these concerns. And I feel empowered by the more trolls that hit me. I mean, of course, that keeps me up at night. And of course, I am worried about it, and I lose sleep over it, and other bad things. But I feel if that happens, I did ask the right questions, and I've asked questions that nobody else dared to ask them. And yeah, there's a feeling of, I think also because I'm backed up by so many people, I feel very empowered to do that. But also because I'm not employed, I don't have any legal team behind me, but I also don't have a boss who tells me, Elise, you should not rock the boat too much, which has happened in the past. I'm a very difficult to employ person. You do not want to hire me because we'll end up with an argument. But I think in this position, that gives me a lot of power to ask the right questions. I don't have any, conflicts or inhibitions from above. And I just feel I'm doing the right thing. But sometimes I have to dig deep, yes.
Wow, this